Welcome to this channel. Today we are going through introduction to neuroanatomy. This is an oversimplified overview, and it's meant for those who are going to start the neuroanatomy course. Today we have 5 different topics. We have the embryonic development of the nervous system. We have the organization of the nervous system. We're going to discuss the neurons and neuroglia, also the reflex arc, and we will wrap it up with the signaling process. So, the embryonic development of the nervous system is generally described as either trimesters or anatomical stages. The anatomical stages is more relevant in our case. Here we have the pre-embryonic period, which lasts about 2 weeks. Then we have the embryonic period, which is 3 to 8 weeks. Then we have the fetal period, which is 9 weeks, essentially 9 weeks up to birth. And it all starts with when an oocyte is fertilized by a sperm. It will form a zygote. This zygote will go through a process called cleavage. Cleavage is a series of mitotic cell divisions, and it will form a mass of cells which is called the morula, and each of these cells is called blastomeres. This morula will move towards the uterus. When it reaches the uterus, it will develop a fluid-filled cavity inside it, which we call a blastocella, and this change will convert the morula into a blastocyst. Then this blastocyst will implant in the endometrium, and when it's implanted, it will consist of an inner cell mass, which we call embryoblast, and this embryoblast will be the embryonic part of the embryo, and it will also have the outer cell mass, which is called the trophoblast, and the trophoblast contributes to the placenta. So the inner cell mass will differentiate into something we call a bilaminar disc, and this bilaminar disc has 2 layers. It's the epiblast layer, which is the top layer, and it consists of columnar cells. Then we have the bottom layer, and it's the hypoblast layer, which consists of cuboidal cells. Then we are going to discuss gastrulation, and the gastrulation is basically when we go from bilaminar disc to trilaminar disc, and it begins to form. It will form a primitive strike along its caudal half. And at the cephalic or the cranial part, we will have a primitive pit. So at the end of this primitive strike, we will have this primitive pit. And the cells of this epiblast will proliferate and migrate through the primitive pit. We go between the epiblast and the hypoblast, and this is a process which we call invagination. And the epiblast becomes the ectoderm, the invaginated cells becomes the mesoderm, and the hypoblast becomes the endoderm. And that's the process of gastrulation. Then after gastrulation, we have another process called neurulation, and this is our main focus today when it comes to the embryonic development. And it is a process where the neural plate involutes, and it will form the neural tube, and the neural tube will make the CNS. And this is the first appearance of the nervous system after the gastrulation process. Also, when someone is pregnant, we can do this CRL, which is a measurement from the vertex or from the cranium until the buttocks on ultrasound, and this is how we can determine the gestational age of the embryo. So we have already discussed the gastrulation. We said that this trimeric disc contains this ectoderm, mesoderm, and endoderm, and something will form from the mesodermal cells, and it's the notochord. And the notochord is it helps us define this longitudinal axis of the embryo, and this notochord will persist and stay and persist and become this the intervertebral disc in the future. But the notochord will release growth factors like fibroblasts, like transforming growth factor beta, so we will form the neural plate. And this occurs in day 17. The ectodermal cells will start to proliferate. This process is called the ectodermal proliferation. It will form this neuroplate, and this neuroplate will thickens, and the embryo will allow this neuroplate to involute. And when it does that, the center of the neuroplate pulls down and the edges roll towards one another, and that's when we get the neural groove and the neural folds. So by day 18, the edges of the neuroplate will move towards one another, like we said, and it will form a neural groove, like a canal, and neurofolds. And at the end of these neurofolds, we will have cells that we call neurocrest cells, and these neurocrest cells are really important because they will develop into different structures, and these different structures will be a part of the peripheral nervous system. So by day 21, we will have the formation of neurotube. At the end of this neurotube, at the ends of it, in the anterior part, we will have a anterior neuropore, and in the posterior part, we will have a posterior neuropore. And they are still open, but we need them to close. And if they don't close, we will have neurotube defects. Folate, which is vitamin B9, is really important in closing the neuropores. So at day 24, the anterior neuropore will close, and at day 26, the posterior neuropore will close. And when this neuropores are completely closed, this neural tube will develop the central nervous system. And something I wrote here is we have the neural tube differentiate into the CNS, and remember the neurocrest cells will differentiate and become parts of the PNS. And some daily remarks: we have day 15 to 17, we have the formation of the notochord. Day 17, we have the ectodermal proliferations, basically the formation of the neural plate. Day 18, we have the formation of the neural groove and the neural folds. Day 21, these neural groove and neural folds will involute and form this neural tube, and the anterior and posterior neuropores would still be open, but at day 24 the closure of the anterior neuropore will start happening or occurring, and then day 26 we have the closure of the posterior neuropore. Then we have something called the vesiculation. So now when we have our neurotube and surrounding it we have the neurocrest cells, the neurotube, we said, is going to become the CNS. We have the cranial part of it which will become the brain and the brainstem, and then we have the caudal part of it which will become the spinal cord. So vesiculation is when the anterior part of it differentiates of this neurotube, and it will form the brain and brainstem. And here we have the primary vesicles. These primary vesicles consist of three different vesicles. We have the prosencephalon, the mesencephalon, and the rhombencephalon, and this is all occurring during week 3. Then when we get to the 5th week, we have these primary vesicles will become the secondary vesicles, differentiate into the secondary vesicles. So the prosencephalon will become the telencephalon and the diencephalon. The mesencephalon will stay the same. The rhombencephalon will differentiate into the metencephalon and the myelencephalon, right? The remaining of the neurotube will stay and become the spinal cord. Then the secondary vesicles will differentiate into more structures. So we said the prosencephalon becomes the telencephalon and the diencephalon. The telencephalon itself will become cerebral hemispheres. The diencephalon will become the thalamus and the pineal gland and the mammillary bodies. The mesencephalon will stay the same through the primary and the secondary vesicles. Then it will differentiate into the midbrain, and then have the rhombencephalon, which divides into metencephalon and myelencephalon. The metencephalon will differentiate into the pons and the cerebellum, and then you have the myencephalon, which differentiate into the medulla. Then we have the spinal cord, which we haven't talked about. So this, if we take a cross section of the neural tube at the cranial part, we will have three zones. We have the marginal zone, mantle zone, and the neuroepithelial zone. The marginal zone will be the outermost layer, and this is where neurons and axons will start forming. The mantle zone is the middle layer, and this is where we find the neuroblast. Then we have the neuroepithelial zone, which is the innermost layer or the most central layer. And from here we have these neuroepithelial cells which will proliferate, and they become the neuroblast. And when they become this neuroblast, they will develop into these neurons and move outwards of the neural tube. When they do that, they will create two plates, alar plate and basal plate. An alar plate will extend dorsally and laterally. It will make the posterior gray horn, and this is where we find the sensory neurons, right? Then we have the basal plate. Basal plate will extend anteriorly and laterally, and it will make the ventral gray horn, and this is where we find the motor neurons. Then we have the neurotube remnants, and the neurotube remnants become the spinal canal, which contains the CSF. So now we have talked about the neurotube, right? We talked about the cranial part of it. We said it becomes the brain and the brainstem. Then we said the caudal part of it, of the neurotube, will become the spinal cord. But surrounding it, we will have neurocrest cells, and these neurocrest cells will become parts. It will become different structures, and this different structure will make up this PNS. So, what does neurocrest cell migrate and differentiate to? This is, for example, the melanocytes. We find that in the skin. This it produces skin pigments like eomelanin, pheomelanin. Then we have these enterochromaffin cells. These chromaffin cells are found in the adrenal medulla. They secrete epinephrine and norepinephrine, and they are important for our stress response. Then the neurocrest cells will differentiate into also the submucosal or this Meissner's plexus and this myenteric, or also called Auerbach plexus. They are important. They are a part of the enteric nervous system, and they are important in regulating the gastrointestinal tract. They will make up the dorsal root ganglia, which is pseudounipolar cells located outside the spinal cord. The collateral and the chain ganglia, which is part of the sympathetic nervous system, the leptomeninges, which is the arachnoid mater, the pia mater, not the pachymeninges, so not the dura mater, because it's derived from the mesoderm. Then also some cranial nerves like 5, 7, 9, and 10, which will innervate the pharyngeal arches. We're going to talk about it more when we talk about the modalities, and some parts of the head and neck, some structures of the head and neck, like muscles and bones. It will also form the ventricles, the ventricular system, and it's from the neurocell of the neurotube. We have the lateral ventricles, the ventricle 1 and 2. They sit in the cerebral hemisphere, and each one of them has a cornu frontalis, like a frontal part, a corpus, a body, a occipital horn, and a temporal part also. They will, these lateral ventricles will communicate with the third ventricle, which sits in the encephalon, and they do that by interventricular foramen of Monro. And this third ventricle will connect with the fourth ventricle, which is through the cerebral aqueduct of Sylvius, right? And the fourth ventricle sits in the brainstem, and it's continuous with the spinal canal or the central canal. So, the organization of the nervous system, as we all know, the nervous system has two components. We have the central nervous system, which is the brain, the brainstem, the subcortical structures, and the spinal cord. And then we have also the peripheral nervous system, which contains the spinal nerves and the cranial nerves. The PNS divides into two: the somatic nervous system, which is in our voluntary control, and then we have the autonomic nervous system, which is involuntary control. And this autonomic nervous system has three parts: the sympathetic nervous system, which is generally known as fight or flight, the parasympathetic, the rest and digest, and the enteric nervous system, which is a part of our gut-brain axis, and it communicates with the CNS to link these emotional and cognitive centers with our intestinal functions. And according to the composition of, because the neurons and neuroglia is what makes up the nervous system, and according to the cell composition of the neurons and neuroglia, we can divide it into two: the gray matter, which is 40%, or the white matter, which is 60%. The gray matter, and it consists mostly of neuron cell bodies, and it develops once we reach our mid-20s. It processes information, it releases new information through the white matter, and it allows us to control movement or memory and emotions. Then we have the white matter, which consists of myelinated axons, and it's kind of the pathway or the motorway, and it develops until a person reaches the middle age, and it allows us to exchange information and communication between the different gray matter areas. So the neurons and neuroglia. As we know, the neurons and neuroglia covers most of the nervous system, and this neuroglia is something we call the glial cells also, and this is the supporting cells. And these supporting cells keep or maintain this homeostasis of the nervous system, but they also have a defensive function also. So keep that in mind. And we have four glial cells that we find in the CNS and two that we find in the PNS, and those are, in the CNS we find the astrocytes, oligodendrocytes, the ependymal cells, and the microglia cells, and in the PNS we find the Schwann cells and the satellite cells. But let's first discuss the neurons, and then we can discuss these glial cells and their functions. And this neuron consists of a cell body, which we call soma. You can also call it perikaryon, and it had at least one branch called a neurite. And these neurites, if they relay signals away from the soma, we call them axons. And if they relay signals towards the soma, we call them dendrites. And the soma contains organelles very similar to other cell bodies. We have like the nucleus, we have the mitochondria, we have the lysosomes, and we also have something called the Nissl bodies, which is the rough endoplasmic reticulum, very prominent in the neurons, and we're gonna discuss about it. So then the cell body, like I said, we can call it soma, perikaryon, cyton, and it basically receives gradient potentials through receptors it has on its membrane, exactly like dendrites, right? And it synthesizes proteins, which is it's necessary for the cell function. They basically make different types of proteins, like neurotransmitters, like enzymes, like membrane proteins. And what's kind of unique here is this neurotransmitter part, right? So we said that if we have a neurite which relays signals towards the soma, we call it a dendrite. So what is a dendrite? Is extensions of the cell body of the soma, and it has receptors, neurotransmitter receptors at its synapse, and it contains different types of channels and receptors. We have one of the channels, these ligand-gated ion channels, which are also called ionotropic, and they are basically quick response channels, and the neurotransmitter will bind to the allosteric sites, and this will open the channel. So the allosteric site, not the active site. Then we also have receptors, which is it's a slower response. It's called metabotropic if it's used these G protein coupled receptors, and this neurotransmitter will bind to the receptor. It will activate the G protein, and it's the same as other G protein. The G proteins kind of works the same, and depending on if it's G stimulatory or inhibitory. But it basically it has this subunits like alpha, beta, and gamma, and this alpha will it's first binded to GDP, and it will release it and bind to GTP, and this subunit will go and activate this membrane enzyme, this adenylyl cyclase, and this adenylyl cyclase will convert ATP to cyclic AMP and so on and so forth. In the end, it will phosphorylate different membrane channels, and this will cause them to open. And when they open, we will have an influx or an efflux, and it can be either anions or cations. If it cations is positive ions, it will lead to an EPSP, the excitatory postsynaptic potential. This EPSP will cause depolarization. This voltage will rise above the RMP, and it will reach the threshold potential. Then we will initiate an action potential, or we can have an influx of negative ions or efflux of positive ions. Either way, it will lead to something we call IPSP, and this will hyperpolarize the cell, right? And this will make it harder for us to initiate and conduct an action potential. So what is the main function of the axons? We said if we have a neurite that relays signals away from the soma, that's what we call an axon. An axon, its main function is basically this action potential conduction, right? We just talked about it. We said the positive ions comes into the cell, making the cell more positively charged. So this EPSP, we will reach the threshold potential, which is minus 55 millivolt. This will open sodium channels, voltage gated sodium channels. We will have an influx of sodium. This will further depolarize the cell. We will reach plus 30 millivolt. When we reach plus 30 millivolt, this will open up or activate the voltage gated potassium channels, and then we will have this potassium will efflux, so it will leave the cell, and when it leaves the cell, it will make the cell more negatively charged. It will repolarize the cell, and these waves, whether they be the depolarization wave or repolarization wave, they travel from the axon helix towards the axon terminal, right? So the axon has two types of transport we need to talk about, and it's how it transports molecules between the soma and the axon terminal, and it does that by microtubules. Here we have two different types of transport. We have anterograde axonal transport, and we have retrograde axonal transport. Anterograde is going from the soma to the axon terminal. Here we use motor protein. Motor protein is called kinesin, and this will transport proteins in vesicles like neurotransmitters, enzymes. It will transport also mitochondria, because we need a lot of mitochondria in the axon terminal, and reactivated pathogens like shingles, because like you see here in retrograde axonal transport, you have, for example, the varicella zoster virus, which causes the chickenpox, and then it can be dormant or latent, and then it can be reactivated as we get older, and it will cause shingles. So another way of transporting is the opposite, is going from the axon terminal to the soma, and this is called the retrograde axonal transport. And here we use another motor protein, it's called dynein, and the retrograde axonal transport is important in recycling or degrading vesicles or mitochondria, or even for communication through these nerve growth factors, because when some parts of the neuron is damaged, it will stimulate a repair, or it will stimulate growth. So we will transport these nerve growth factors via the retrograde axonal transport. But also pathogens, we already talked about varicella zoster, but you have here polio, rabies, and herpes simplex. So the axon terminal is the distal part of the axon, and the function of the axon terminal includes a neurotransmitter release and neurotransmitter reuptake. And when we talk about the neurotransmitter release, here we already talked about the depolarization of an action potential. This will reach, eventually it will reach plus 30 millivolt. It will reach just 30 millivolt. When it reaches 30 millivolt, it will activate voltage gated calcium channels. And then we will have an influx of calcium, and this calcium will facilitate a fusion between these neurotransmitter containing vesicles and the cell membrane. So these vesicles have proteins called V-SNE proteins on their membrane, and the cell membrane has proteins called the T-SNE proteins. Like we said, calcium facilitate the fusion between the two. V-SNE proteins are synaptobrevin and synaptotagmin. The T-SNE proteins are syntaxin and SNAP25. And when the calcium facilitates this fusion, this will lead to a neurotransmitter release into the synaptic cleft via a process called exocytosis. Then we have a neurotransmitter reuptake. That's another function of the axon terminal. When we have passed on the axon potential to the postsynaptic neuron, we need to remove the neurotransmitter. Obviously, we don't want it to stay there and keep stimulating. So we can do that by two ways. We can either degrade it, right, or we can reuptake it. So if we degrade it, we have enzymes on the cell membrane or the synapse itself, which break down these neurotransmitters, or we can reuptake it and bring it back to the axon terminal so we can use it once again. Then we need to discuss the types of axons. And here we have three types. We have the axosomatic, the axoaxonic, and the axodendritic. The axosomatic, this is very self-explanatory. So the axosomatic ones will be between the axon and the soma. The axoaxonic will be between these two axons, and it's for this all or nothing phenomenon. And then you have axodendritic, which is between the axons and the dendrites. Right, myelin. So we said that we have these neurites, which two types of neurite. We have these axons and we have these dendrites, and they have a protective layer, or let's call it a covering. And this covering is called myelin, and it's made of proteins and it's made of lipids, and basically acts like an insulator, electrical insulator. It increases the speed of conduction, so it helps to increase the transmission speed. And it's made of glia cells, which wraps around the neurite, and they will squeeze out their cytoplasm towards the periphery, and they will leave only their cell membrane in the inner layer. And this is how we have this. This is how we create this myelin sheath, and it's not covering the whole span of the neurite. Remember this. In the axon, we have gaps, and these gaps is what we call nodes of Ranvier, right? And we're going to talk about the nodes of Ranvier, but in the CNS we have the oligodendrocytes, which synthesize myelin, and they synthesize several myelins. But in the PNS we have the Schwann cells, which synthesize the myelin, and they can usually only do one at a time compared to oligodendrocytes. So we just mentioned the function of the myelin, and it helps to conduct these electrical signals faster, and we do that via a process called saltatory conduction compared to unmyelinated axons, because they use another process called continuous conduction, and that process is much slower compared to saltatory conduction. And basically what that means is in myelinated axons we have gaps where we don't have any myelin, right? And these gaps we call them nodes of Ranvier, and in these nodes of Ranvier we have voltage gated sodium channels. So if the first one will be activated, we will have an influx of sodium. This influx will depolarize the cell. This depolarizing wave will go to the next node of Ranvier, where we have the second voltage gated sodium channel, and it will activate this sodium channel. Then we will have another influx of sodium, and this depolarizing wave will go to the third node of Ranvier, and so on and so forth, compared to a continuous conduction, where we don't have any nodes of Ranvier because we don't have any myelination, and therefore we will have a slower. So I want you to see saltator conduction as the depolarizing wave will like jump from the first node of Ranvier to the second, then to the third. So in that way it goes faster compared to continuous. Then we have the conduction velocity, and it's dependent on, we can classify it upon if they have a myelination and their target organs, or we can classify them based on their diameter and their transmission speed. And if we classify them based on their myelination and target organ, we have three. And here conduction velocity will be based on the degree of myelination or the diameter of the axon. So we either can classify them in just group A, B, and C, and group A will have the fastest conduction because they are the most myelinated, they are somatic, both afferent and efferent. Then you have group B, which is moderately myelinated, and they are vegetative preganglionic. Then you have group C, which are called the Remark fibers, and they are unmyelinated axons, so therefore they will have the slowest conduction velocity. And they are also their target organs are the vegetative postganglionic. The conduction velocity, we said it depends on the diameter, so here it's very obvious that the larger diameter we have, the faster conduction we will have, and the smaller diameter, the slower conduction. And like I said, we can classify them also based on the transmission speed. Here we have type 1A fibers with a diameter of 12 to 20 microns. The conduction rate is between 70 and 120 meter per second. They are useful in carrying motor signals to skeletal musculature, and they relay proprioception from these muscle spindles. Then we have type 1B fibers. They are they have smaller diameter of 10 to 15 microns, and their conduction rate therefore will be slower, so it will be around 60 to 80 meters per second. And they are involved in the afferent relay from the tendons and the skin. Then you have type 2 fibers, which has a diameter of 5 to 15 microns, conduction rate 30 to 80, and they carry this sensory from Meissner's and Pacinian corpuscles, which we find in the skin, but also proprioception from muscle spindles. Then you have A gamma fibers and A delta fibers, and both of them are 3 to 8 microns in diameter, but the A gamma fibers have a less or a slower conduction rate between 10 to 30 meters per second compared to the A delta fibers, and both of them carry efferent information to the muscle spindles, and they carry also temperature and pain from nerve endings. And then we have type B fibers. They are the smallest in diameter when it comes to the myelinated fibers, and they have a conduction speed between 5 to 15, and you can find them in the white rami of the cranial nerve 3, 7, 9, and 10. Then we have type C fibers. This is the fibers. This is the Remark fibers. They don't have any myelination. They are very, very small in diameter. Therefore, they will have a very slow conduction rate, and they carry nociception, and temperature and olfactory afferents. Then we have types of neurons, and this type of neurons, they can be of different type. So we can classify them as multipolar, bipolar, or pseudo-unipolar. The multipolar ones are the most common types, most common type you're gonna find in the CNS, and they form the autonomic ganglia, which is part of the PNS. They have one single axon, and they have symmetrically radiating dendrites. They can also further be subclassified based on their branching pattern of their dendrites or the shape of their soma. And we can classify them into stellate, pyramidal cells, fusiform, Purkinje, and glomerular cells. So if the first three, the stellate, pyramidal, fusiform, you're gonna find them common or abundant in the cerebral cortex. Purkinje cells, you're gonna find them in the middle layer of the cerebellar cortex. And then you're gonna have these glomerular cells, which we're gonna find in mostly in the olfactory system. So stellate, they are star-shaped. They are isodendritic, something we call isodendritic, and it basically means that dendrites have equal length, and they radiate uniformly in all direction. They are found in the spinal cord, formatio reticularis, and cerebral cortex. We find them in the layers 2 and 4 in the cerebral cortex, and they are classified as Golgi type 2 fibers. And Golgi type 2 fibers basically means, we're gonna talk about it, but it basically means short axons. Then you have these pyramidal cells that has a pyramidal shaped soma, and they have a single apical dendrite that ascends towards the cerebral cortex. They are most abundant in the cerebral cortex. We find them also in the hippocampal gyrus. In the cerebral cortex, we find them in layers 3 and 4. And when we talk about BET cells, basically means pyramidal cells in the motor cortex. If we talk about Maynard cells, we are talking about pyramidal cells in the visual cortex. Then we have these fusiform cells, and they are spindle-shaped. They have a spindle-shaped and flattened soma, and they have dendrites on the both ends. Find them in the 6th cerebral cortex layer. Then we have these Purkinje cells. They are flash-shaped. They have a flash-shaped soma, and on the apical side we have these tree-like dendritic branches. We find them in the cerebellar cortex, and they are motor neurons that project long axons. Then we have these glomerular cells, which have convoluted dendritic branches, and they have this. They can either be mitral cells or tuft cells. Mitral cells is because they have a soma that resembles a bishop's mitre, and we can find them mostly in the olfactory bulb. So, like I said, on the basis on the axonal length, multipolar neurons, we can categorize them into Golgi type 1, and that's with long axons, or they can be classified as Golgi type 2, and they have short axons, and they usually inhibit neighboring neurons, and we call it the lateral inhibition. Then bipolar neurons also do the lateral inhibition, but they also do reciprocal inhibition. And reciprocal inhibition, it basically means I inhibit you and you inhibit me. And they have one axon and one dendrite, and this both extending on the opposite poles. And here the bipolar neurons, you can find them more in the special senses like retina and the olfactory system. Then you have these pseudo-unipolar neurons. Unipolar neurons are found only in invertebrates, but we have pseudo-unipolar neurons instead, and they have two processes from the soma, one from the soma, two processes, so one peripheral process and one central process, and the peripheral one coming from the periphery, the central one going towards the CNS. And those the pseudo-unipolar neurons are relay neurons, so interneurons found in the dorsal root ganglia and the trigeminal ganglion. Then we have something called anaxonic cells, and we can find them in the retina. We call them amacrine cells in the retina, but in the olfactory bulb they are known as granule cells. So neurons, we can classify them according to their function. We can classify them as sensory neurons, interneurons, or motor neurons. We can also classify them according to their order. So if they are first order neuron, that's where the impulse starts. Second order neuron, they are relay neurons or interneurons. And third order neurons are neurons going to the effector organs. And this is commonly more found in the ascending system, where we have this three neuron circuit. But in the descending system, we find it more. We commonly find more two-order system, and a more stationary term for this is the upper motor neuron and lower motor neuron. Then we have to talk about neuroglia. And neuroglia, as we mentioned before, they are supporting cells for the neurons, and we can find them both in the CNS and the PNS. We have already mentioned that in the CNS we have these astrocytes, oligodendrocytes, epidermal cells, microglial cells, and in the PNS we have the Schwann cells and the satellite glial cells. And if we're going to start with the CNS, we can start with the astrocytes. Obviously they are only found in the CNS, and they have these food processes that extends from the soma, very characteristic. In the white matter we call them fibrous astrocytes. In the gray matter we call them protoplasmic astrocytes. And their function is, they have four functions, and these are being a component and modulating this blood-brain barrier. Then they have also potassium buffer function, and they also remove excess neurotransmitters, and they can act like glycogen storage. So blood-brain barrier, we have to talk about the components of the BBB, and if we talk about it, we're gonna mention it from the inner to outer, and the outer meaning towards the neural tissue. So the first layer is the endothelial lining of the blood vessels. Here we have tight junctions located between these endothelial cells. They help reduce the permeability across this blood-brain barrier. Then we have the basal lamina, and it's a connective tissue. Then you have the astrocytes, which they have these food processes also, and around it you can find these pericytes. Then the function of the blood-brain barrier, we regulate what leaves the blood and what enters the neural tissue. So substances that can just diffuse passively is oxygen and carbon dioxide, also lipid-soluble substances. But we have, if they are water-soluble substances, then they require transporters, or if they are electrolytes. Substances we do not want is obviously proteins, because they can act like neurotransmitters and it will have an undesirable effect. So what does astrocytes have to do with this blood-brain barrier? It releases growth factors. These growth factors will stimulate these endothelial cells. These endothelial cells will express more tight junctions. When they do that, they will reduce the permeability of the blood-brain barrier. Therefore, the BBB becomes more selective. Then we have regions in the brain where we don't have any BBB, and this is the circumventricular organs. Here we can find area postrema, because it's area postrema triggers our vomiting reflex, right? So it needs to be absent of this blood-brain barrier, so it can be sensitive towards toxins and drugs. Then we have these osmoreceptors. We can find them in the hypothalamus, and they need to sample the osmolality of the blood and then trigger either vasopressin release or activate our thirst mechanism. Here, two locations, we have the subfornical organ and the organum vasculosum of lamina terminalis. Then we have this hypothalamic pituitary axis, the HPA, and it basically, the hypothalamus will secrete hormones which go on and stimulate the cells in the pituitary gland so they can further release hormones and we can maintain our homeostasis. And it uses the bloodstream as a communication pathway. I know I promise we're not going in the depth, but I needed to mention something I find very interesting, and it's Claudin 5. It's a protein in the tight junctions of the blood-brain barrier, and we have seen an increase in patients who are suicidal, and in the future it can be a potential biomarker of suicide risk, but obviously more research are needed. Then we already mentioned that the astrocytes have this potassium buffer function, and when we go through the repolarizing phase of the action potential, this will open up these voltage-gated potassium channels, and we will have an efflux of potassium. During rest, we have these leaky channels that can that lets the potassium to exit. And if we have too much, we will have the sodium-potassium ATPases, which will try to push potassium back into the neuron, but they don't have such a big effect. And we're going to talk about it when we talk about the signaling process. But why is this a problem? Yeah, well, obviously, if we have a high concentration of potassium outside the cell, this will affect the repolarizing phase, and it will affect the action potential during resting membrane potential. So how do we resolve this? Well, thankfully we have these astrocytes, and they can express specialized transporters, potassium transporters on their membrane, and they have a gap junction between them, so they can effectively distribute this potassium. So they can pick up the potassium and they can distribute them among each other. And this is how they work as a potassium buffer. Then another function of astrocyte is removing excess neurotransmitters. So glutamate, as we know, is the most common excitatory neurotransmitter, and it releases from the presynaptic neuron. It will bind to this ligand-gated ion channels on the postsynaptic neurons, and it will have its stimulatory effect. But after it has exerted its effect, it will dissociate from the receptor, and then we reuptake them via these glutamate transporters back into the presynaptic neurons in the terminal bulb. Sometimes this glutamate transporter becomes saturated, so then we will have too much glutamate sitting on the synapse and exert a longer effect. So what does astrocytes do about this excess glutamate? Well, they express another transporters on their membrane. This time it's glutamate transporters. These glutamate transporters will uptake the glutamate, then inside the astrocyte they will convert it via an enzyme we call glutamine synthase. It will convert this glutamate into glutamine. Now when we have glutamine, it can go two ways. So it can either be taken up by the presynaptic neuron via glutamine transporters, and then once they are in the presynaptic neuron, they will be reconverted back to glutamate. Or when the astrocytes release glutamine, it's taken up by nearby GABAergic neurons, and they will be converted back to glutamate via the glutaminase enzyme, and then from glutamate into GABA via glutamate carboxylase. So it can go two ways, to the presynaptic neuron or to the nearby GABAergic neurons. Then we said it has the fourth function, and it's the glycogen reserve. And when we have a decrease in ATP, the astrocyte will sense this ATP level. And they will have these GLUT1 transporters on their blood-brain barrier, and they make them easier for glucose to cross the blood-brain barrier. Then it will convert it, once in the astrocyte, it will convert it into glycogen so we can have it for storage. And once the neurons have a low in ATP, the astrocyte will break it down to glucose, back to glucose, then to pyruvate, then to lactate. And this lactate will enter the neuron and is reconverted back to pyruvate, and pyruvate will be used to make ATP. Then we have in the CNS the second type of glia cells were these ependyma cells. And they are basically cuboidal cells, and they are found in the ventricles. They are found in the lateral ventricles, third and fourth ventricles, and they are component of this blood CSF barrier, which is found inside these ventricles. And this choroid plexus will form this blood CSF barrier. And if we're gonna talk once again from layers from the inner to outer, is we have first the endothelial cells which has fenestrations. Then you have a basal lamina, and it's made up of connective tissue. And then you have these ependyma cells, and they have tight junctions between them. And this is they play the most crucial role here in the selectivity, in the permeability of the choroid plexus. And the choroid plexus will regulate what leaves and what enters the CSF. And the ependyma cells also have cilia, which helps to move CSF throughout this ventricular system. Then we have microglia, the Ortega cells, and they are found also only in the CNS. They have this multiple dendritic-like extensions, and they migrate from the bone marrow as monocytes, then become permanent residents in the CNS, and are then called microglia. They are the only glia cells with a mesodermal origin. So for example, if you have a pathogen damaging the CNS, this will lead to cytokine release. It will activate the microglia. The microglia will release nitric oxides, other cytokines or chemotactic factors, also reactive oxygen species like hydrogen peroxide or superoxide, but if they release ROS, they can damage other neural tissue, and this can lead to demyelination. But in a normal situation, this microglia will phagocyte these pathogens, express the antigen on the MHC2 complexes, which they have on their membrane, and present it to the CD4 plus cells. This will activate these T cells. They will release more chemotactic factors to attract immune cells, so more immune cells will come to the area and kill the pathogen and clear this cellular debris. Then we have the oligodendrocytes, and we are still talking about the CNS, the glia cells, which we can find in the CNS. This oligodendrocyte, we have already mentioned it because it makes up this, it produces the myelin, and we said that it can produce myelin, several myelins at the same time, so 30 to 60 axons. And this include the myelinate axons of the CNS. This include the brain, spinal cord, and the optic nerve. But here's the thing, they cannot regenerate if they are damaged. So the most common demyelinating disease you're gonna find in the CNS is multiple sclerosis. Now let's talk about the PNS. And in the PNS we have the Schwann cells, which are very alike oligodendrocytes, which we find in the CNS. But here, multiple Schwann cells can only myelinate one axon, and they myelinate spinal nerves and these cranial nerves from cranial nerve 3 up to cranial nerve 12. The most common demyelinating disease we find in the PNS is GBS, the Guillain-Barré syndrome. Here's the thing, compared to the oligodendrocytes, they can regenerate if they are damaged or demyelinated compared to oligodendrocytes. Then we have these satellite glia cells. Older term is the amphicytes. They are only found in the PNS. They are kind of like a similar function as astrocytes in the CNS, so they maintain the homeostasis. But they also have a very similar function as microglia because they are also protective. They have this protective mechanism. And we also have this radioglia cells, and they are basically progenitor cells, which we are which is found in the developing nervous system, and they are a big target of interest for researchers. So, what is a nerve? Well, a nerve is when we have multiple axons bundled together. This will create a structure we call a nerve. And within each individual axon, we have a connective tissue layer called the endoneurium. And the endoneurium creates this endoneural fluid which protects it from injuries. And when we have multiple axons bundled together, they will be bundled into a fascicle. And every fascicle has another connective tissue covering called the perineurium. Then multiple fascicles, so with vasa nervorum, which is blood vessels of the nerve, are enclosed by another layer, and this is the outermost layer, and it's called the epineurium. So these nerve fibers, we can classify them based on their modalities. We have afferent and we have efferent fibers. The afferent ones are, we have 5 of them. We have the general visceral afferent. They carry general sensations from the viscera and the smooth muscles. Then we have the general somatic afferent, which carries general sensations from skin, skeletal muscles, tendons, etc. Then we have the special visceral afferent, which carries senses derived from the endoderm, like the olfactory and the gustatory. And then we have the special somatic efferent, which carries senses derived from the ectoderm, like visual and auditory. Then we have the efferent fibers, and the efferent fibers carries. We have this general visceral efferent, which carries autonomic motor innervation for internal organs, smooth muscles, and glands. Then you have the general somatic efferent, which carries motor innervation for skeletal muscles. And then you have this special visceral efferent, which carries motor innervation for muscles derived from the pharyngeal arches. And we mentioned the pharyngeal arch, but I didn't mention that we have 6 of them, and the 5th one exists only during embryonic period, so we will not have any structure resulting from it. So the trigeminal nerve carries muscles derived from the first pharyngeal arch. We have the muscles of mastication. Here we have the anterior digastric, the tensors, and then we have the cranial nerve, nervus facialis, which carries signals from pharyngeal arch 2. Here we have muscles of facial expression, posterior digastric, we have stylohyoid, and then we have the cranial nerve 9, the glossopharyngeus, which carries motor innervation for muscles derived from the pharyngeal arch 3. This is typically the stylohpharyngeus, and then we have vagus, which carries motor innervation for muscles derived from pharyngeal arches 4 and 6, and these are the muscles of pharynx and larynx. And then we have something called the reflex arc, and the activity inside the nervous system is mainly based on something we call the reflex act, and it's a response of the CNS and the whole organism when it's subjected to different stimuli, and this phenomenon is called the reflex arc. So it is both found in the CNS and the PNS, and it has 5 components. First component is the receptors, the second is the efferent pathway, third reflex center, then the efferent pathway, and last but not least the effectors. So the receptors are specialized cells or non-neural tissue which are wrapped around these nerve endings, and they are designed to detect the stimuli that excites them, and they will transform this energy into electrical signals. And we can classify them in 3. We can classify them as exteroreceptors, proprioceptors, or interoreceptors. And the exteroreceptors, we find them in the periphery or the surface of the body, and they perceive stimuli from the environment like touch, pressure, temperature, nociception. And then we have the proprioceptors, which are located in our osteomuscular system, and they offer mainly the position of the body, the direction of various movements, and also the muscle tone. And then we have the interoreceptors, which are our visceral receptors of our internal organs and also blood vessels, and they perceive visceral sensitivity and activity, and they contribute to the supervision of homeostasis. And these receptors can be activated in two ways. Either they can be tonic receptors, so they will continually fire, and this is, for example, the proprioceptors, or they can be phasic receptors, so they only fire when there is a change in the environment. A prime example of this is thermoreceptors. Then we have the adaptation of it. Some say the inactivation. You're gonna find it in some literature naming it the inactivation of receptors, but they are not inactivated. They will rather adapt. So in adaptation, they can either be rapidly adapting or they can be slowly adapting. And if they are rapidly adapting, the receptor very quickly stops firing if there is a continuous stimulus. A very good example of this is touch receptors on our surface. Here, if we put on clothes, you're not going to think about these clothes after a few seconds because the receptors are rapidly adapting and they're going to stop firing. Then we have the slowly adapting receptors, and they will, like the name suggests, they will maintain firing under continuous stimulus, and they are typically muscle stretch receptors found as proprioceptors. Then we have something called the receptive field, and the receptive field is what one receptor is monitoring, and the receptive field is the area monitored by one receptor. So if we touch anything in that field, the sensation will come from the whole receptive field because we have one receptor monitoring it. And here we have a large receptive field. This is not that specific. They have very low receptor density, very poor localization. A good example of this is the skin on your back. And then we have these small receptor fields which have a higher receptive density. You can localize it very specifically because they have obviously a higher receptor density. A good example of this is the skin on your fingertips. These receptors can either be unencapsulated or they can be encapsulated. And the unencapsulated are 3 different receptors. We have the free nerve endings, we have the tactile disc, commonly known as Merkel disc, and we have these here follicle receptors. The free nerve endings, they have There are exterior receptors, interior receptors, and proprioceptors. Their stimulus type is thermo, chemo, mechanoreceptors, and even nociceptors. We find them in most body tissues, and they are most dense in connective tissues, and we can find them in the epithelia also. Then we have this Merkur disc. They are exterior receptors. They have their stimulus type is mechanoreceptors. They are very slowly adapting compared to the hair follicle receptors, and they are based in the basal layer of the epidermis. Then you have these hair follicle receptors. They are also exterior receptors. They are also mechanoreceptors, but it's not light pressure, it's hair deflection, and they are rapidly adapting. And we find them surrounding the hair follicles. And like I said, we have these 6 encapsulated receptors. We have the Meissner corpuscles, we have Pacinian, Rufinis, Massospindos, GTO, and the joint kinesthetic receptors. The Meissners are exterior receptors. Their stimulus type is mechanoreceptors. Here we have light pressure, discriminative touch, and low frequency vibration. They are rapidly adapting. A difference between Meissners and Pacinian is that when it comes to the stimulus type is very similar, but here it's more stronger type of stimulus, like a deep pressure or a high frequency vibration. But the Meissners, we find them in dermal papillae of the hairless skin, like nipples, like fingertips, like the soles of the feet. Pacinian corpuscles, we also call them Vater Pacini, very similar to Meissners, and we also find them in the dermis. We find them everywhere, but they are very abundant on the fingers. Then you have these Rufinis endings, and they are exterior receptors, proprioceptors. Their stimulus type is similar to Pacinian by having this deep pressure, but they are more slowly adapting. And we also find them in the dermis, and we can also find them in the joint capsules. Then we have these Massospindos, which are proprioceptors. Their stimulus type is mechanoreceptors, and they are found in the skeletal musculature. Then we have the GTOs. They are proprioceptors. Their stimulus type is when we have stretch of the tendon or we have a tension. Their location is the tendons, like in the name. Then we have the joint kinesthetic receptors. They are proprioceptors. The stimulus type is also mechano, but they have nociceptions. Also, they are found in the joint capsules of the synovial joints. Then, so we talked about the reflex, or we talked about the receptors, right? So we said the next thing was this afferent pathway, and this afferent pathway is the somatic and visceral sensitive fibers of the peripheral nerves. Here we have we can find different types. Type A fibers, they start from the tactile corpuscles in the skin, but also the free nervous endings. Type C fibers start from the free nervous endings we find in the skin, and they are more nociceptors. Type 1 and 2 fibers, they originate in the neuromuscular and neurotendinous spindles, but we can also find them in the articular corpuscles. Type 3 and type 4 fibers originate from the muscular and the articular nociceptors. So now we have talked about the receptors and then the afferent pathway. Now we come to the reflex centers, and here is where we determine the complexity of the reflex, and we can classify them as two. We either have these monosynaptic reflexes or polysynaptic reflexes, and a monosynaptic reflex is made up of two neurons. It has a single synapse between them. A prime example of this is the patella reflex. Polysynaptic reflexes are excitatory or inhibitory relay neurons with several synapses between them. A good example of this is the postural reflexes, and the reflex centers can be found in three levels. The medulla level, here we find both somatic and visceral reflexes. And then you have the subcortical level. Here we find arcs, reflex arcs in the brainstem, cerebellum, the encephalon, basal ganglia, and then you have the cortical level. Then we have the efferent pathway, which is made up of the descending pathways consisting of both CNS and PNS components. Here we have the type A fibers. They can be alpha motor neurons, which is outside this neuromuscular spindle, but also gamma motor neurons for the spindle fibers. Then you have the type B fibers, which are preganglionic fibers on the vegetative system, and type C fibers, which are postganglionic fibers on the smooth muscles, cardiac muscles, and glands. Then, according to their function and their destination, these efferent fibers can either be skeletal-motor, motor for the spindles, or vegetative. The skeletal motor will innervate the non-spindle skeletal musculature. Then you have the motor for the spindles, and 30% of these motor fibers will modify the length so they can maintain this its tonus. Then we have the vegetative, which are preganglionic and postganglionic fibers for the cardiac muscles. We call them cardiomotor. Then we have this visceromotor, which is for the smooth muscle fibers in the internal organs. Then you have the vasomotor, which is for the smooth muscles in the vascular walls. Then you have the pilomotor for the erector muscles of the hair follicle, pseudomotor for the sweat glands, and for the digestive gland we have the secretomotor. So now we have talked about the receptors, the afferent pathway, the receptor centers, and the efferent pathway. Last but not least, we have these effectors who are connected with the efferent pathway through a special synapse we call neuroeffector synapse. And it can either be neuromuscular or it can be neuroglandular. And this neuroeffector synapse is very similar to a regular synapse. So we have synaptic buttons, we have a synaptic cleft and a postsynaptic component, and it works similarly like other synapses. So it has these messengers, whether they be neurotransmitters, hormones, and then you have they will be inactivated and recovered by the postsynaptic neuron, and based of their stimulus they can be excitatory or inhibitory in this reflex act. Functionally, we have three reflexes, and they can be either protective, adaptive, or orientational. The protective reflexes are protecting us from harmful influences and is associated with pain, and they can either be somatic, which is for like coughing and sneezing, or they can be vegetative or combined vegetative, and they remove noxious substances from the body, for example like vomiting. Then we have these adaptive reflexes, which are usually polysynaptic, and they work as a servo for setting this sensibility of receptors, or they can dispatch this right amount of motor commands among somatic effectors, for example like stance. Then we have orientational, and they will cooperate with several reflex centers, and the aim here is to gather the best quality of input. Therefore it's going to work with several reflex centers. Yes, and then we have the last topic, which is the signaling process. And the signaling process is consisting of three components. We have the resting membrane potential, we have the graded potentials, and then we have the action potentials. And the resting membrane potential is basically when a neuron is at rest, and the average voltage here is around minus 70 millivolt to minus 90 millivolt. And here we have three type of channels, and they maintain this resting membrane potential. The first one being the sodium potassium ATPases. They push 3 sodium ions out and pump 2 potassium ions into the cell. and they require ATP. You have it in the name, it requires ATPase because they push ions against their concentration gradient. We all know that sodium is higher outside the cell and the potassium is higher inside the cell. So it will be against its concentration gradient, therefore it will need ATP to work. So we also have leaky potassium channels, and inside the cell this potassium will interact with anionic proteins like phosphates, like amino acids, and they will move. These channels are always open, so they will allow potassium to freely cross the cell membrane down its concentration gradient because it's in high concentrations inside the cell. But when it leaves from inside the cell to the outside of the cell, these anionic proteins will create an electrical gradient between them and the potassium, and therefore making it want to trickle back into the cell. But as potassium leaves the cell, these interactions with these anionic proteins will create an electrical gradient, and this will make the potassium want to trickle back into the cell. And when we have this electrical gradient plus this chemical gradient, we make the electrochemical gradient. And this bidirectional movement will keep going until we have no net movement across the cell membrane. So no potassium leaving and no potassium coming into the cell. And this is what we call the equilibrium potential for potassium. Then we have this leaky sodium channels, and they will allow the sodium to trickle into the cell, so down its concentration gradient, because we know that sodium is in higher concentrations outside the cell, and the neuron is 90 to 100% more permeable towards potassium and not sodium. Now let's talk about the graded potentials, and the graded potentials are the sum of the EPSPs and the IPSPs needed to approach the threshold potential in the axon helix so we can initiate an action potential. And the EPSPs is excitatory postsynaptic potential, so small depolarizations, so we can bring the RMP, the resting memory potential, closer to threshold. The opposite of it is the IPSPs. Here we have small hyperpolarizations to bring the RMP further away from the threshold potential, so it's harder for us to initiate an action potential. And it works by having these channels which are involved in the graded potentials. So these ligand-gated ion channels, they can either be glutamate sensitive channels or GABA sensitive channels, and if they are glutamate, like we mentioned before, glutamate is a stimulatory neurotransmitter, most commonly found stimulatory neurotransmitter, and it binds to the receptor, it opens the channel, we will have cation ions influx, or positively charged ions. This will depolarize the cell and it will bring it closer, it will bring the RMP closer to the TP. The opposite of it is the GABA sensitive channels. So we already know the GABA is the most common inhibitory neurotransmitter, and when it binds to the receptor it opens the channels. Here we have anion influx, or we can also have cation efflux. Either way, this will hyperpolarize the cell and it will bring the RMP further away from the threshold potential. And we can do that by two types of ways: temporal summation or spatial summation. Temporal summation is basically one presynaptic neuron. It attempts repeatedly to stimulate, or it can also inhibit one postsynaptic neuron. See it as nagging. So temporal summation is a presynaptic neuron nagging on the postsynaptic neuron. And when the EPSP is greater than IPSP, this will make the cell this will depolarize the cell, and we can initiate and conduct an action potential. Or we can do that by spatial summation. Spatial summation is not through a nagging process. It's more kind of simultaneously you have multiple presynaptic neurons and they try to stimulate or they try to inhibit the postsynaptic neuron. And once again, when the EPSP is greater than the IPSP, this will depolarize the cell and bring the RMP closer to the threshold potential. And just very short clinical significance, some patients have seizures. This is non-synchronizing and excessive electrical activity in the CNS. When someone having a seizure, we can give them IV a group of medication called benzodiazepines. One of them is the lorazepam, which is a GABA agonist. This lorazepam will cause an anion influx. This will hyperpolarize the cell, bring it further away from the threshold potential. This will help us to abort this seizure. And then last but not least, we have the action potentials, and it's the electrical activity conducted down the axon. Once we reach threshold potential in the axon helix, to reach this TP, we need to generate an action potential at approximately minus 55 millivolt. The channels involved in the action potentials are two channels. We have the voltage-gated sodium channels and we have the voltage-gated potassium channels. The voltage-gated sodium channels have two gates. We have the activation gate located in the outer aspect of this channel, and then you have the inactivation gate located in the inner aspect of this channel. And they span the whole length of the axon. And when we reach minus 55 millivolt in the axon helix, the activation gates will open up and they will allow the sodium ions to enter the cell. The inactivation gates will slowly start closing simultaneously. And when we reach our peak at approximately plus 30 millivolt, the activation gates will still be open, but the inactivation gates will close and stop further entry of sodium. Then when the voltage reaches the resting memory potential at around minus 70 millivolt, the activation gates will close, the inactivation gates will open, and we are at a position where we can be stimulated once again. So the other channel that is involved in action potentials is this voltage-gated potassium channel. And it opens up at plus 30 millivolt when we reach our peak depolarization voltage, and it will allow potassium ions to leave the cell and cause the cell to become more negatively charged until we reach our resting membrane potential, which is around minus 70 millivolt. When we reach our RMP, that's when these gated channels close and they won't allow any more efflux of potassium, but they take a little bit time. They are very slow in closing. That's why we can get our overshoot or this hyperpolarization. Before we move on, I know I said we're gonna keep it as an oversimplified overview. I just wanted to mention there is this NAV 1.8 inhibitors currently going through phase 3 trials. NAV 1.8 is tetrodotoxin resistant voltage gated sodium channels, which we find in the dorsal root ganglion, and it's a new pain medication. Is very promising and is better tolerated compared to the classical ones. Then we have this absolute and relative refractory periods. This is periods where we can stimulate or not stimulate another action potential. When it comes to absolute refractory periods, we cannot, it doesn't matter how strong stimulus we have, we cannot at this time, during this time, stimulate another action potential. And this is the time between the peak of depolarization until we reach the RMP. When it comes to the relative refractory period, during this time, if we have a strong stimulus, then we can initiate a new or generate a new action potential. And this is the time between the hyperpolarization until the RMP is reclaimed once again. In the end, we have the secretory stage of the action potential. When this depolarizing wave travels down the axon towards the axon terminal, this will activate voltage-gated calcium channels. And these voltage calcium channels are located in the axon terminal. and we will have an influx of calcium. It's usually when the voltage is around plus 30 millivolt. When calcium rushes into the axon terminal, this will, we have mentioned it before, it will facilitate a fusion between the vesicles and the synapse membranes. This fusion will lead to neurotransmitter release through a process we call exocytosis. When the neurotransmitter has been released, right after that we will have this repolarizing wave moving down the axon terminal, and the return of the RMP will inactivate the voltage gated calcium channels. This will prevent further entry of calcium. This will prevent further exocytosis of neurotransmitters, and it allows the axon terminal to reset and recycle these neurotransmitters and ready to go for the next action potential. Thank you for watching. I hope you enjoyed the video, and if you did, please leave a comment, like, or even subscribe. Thank you so much.